Good afternoon, everyone. So here is Weihang, and today my topic is maybe a little bit interesting. So it's about inferring epigenomic information of spatial transcriptomic data using the single-cell multi-omics reference. So here, so because there are already many introductions about the single-cell omics data or the spatial transcriptomic data, so I will not make no further introduction about such kind of data. So we will just go straight forward to our topic. So firstly, why inferring the epigenomic modality for the SD data is important? Because we know that given a spatial transcriptomic data, we can get the spatial gene expression information. However, we know that only the spatial gene expression information is not enough for us to investigate the spatial gene regulatory mechanism. So that's why we want to try to find some solution to add some new information to one given ST data. So here we want to just add some epigenomic information for given ST data. So in this way, we can just investigate the region-specific epigenomic patterns, for example, some spatial chromatin accessibility pattern or some histone-modification. And we can also check the region-specific, for example, the TF binding site motif enrichment, and then we just combine this kind of information with the spatial gene expression information, and then we can make a more comprehensive understanding for this spatial data. And here is one example I give to you to show how the additional information can help us. So the left one is just the clustering results based on the one spatial gene expression patterns. So we can see that if there are two clusters, and for the spatial gene expression, it's hard to just separate these two clusters. And if we just use for example, the spatial peak accessibility pattern, so we can very easy to just separate these two clusters. So that's why this kind of epigenomic information can help us to better investigate a given SD data. And so the question is this possible to just inferring some epigenomic information for given SD data? So the answer is yes. So we use the single-cell multi-omics data as reference. And we also develop an encoder-decoder-based graph neural network model. So here is a very simple illustration of the framework. So the input is just, as we have talked about, is one paired single-cell multi-omics reference. And then the other one is the spatial transcriptomic data without the epigenomic information. It's just a very simple SD data. And then we use this kind of two data to generate three different kinds of graph. And these three graphs, they just have some different information. And so then we can input these three graphs into one graph fusion encoder to just merge this information to a shared embedding space. And in this shared embedding space, we can have both spots from the SD data and the cells from the reference data. And then they are just merged with each other based on their similarity. And then we can feed this shared embedding space to another statistical decoder. And then we can just generate or just infer the epigenomic part for this given SD data. And here is the detail of our model. So just as I have mentioned, that the input consists the SD data and the single-cell multi-omics data. And after we get this kind of two data, we just construct the three kind of graph. We can see that it's the reference graph and the reference and SD graph and the SD graph. And then we just feed these three graphs into, based on the heterogeneous graph learning, we can just merge these graphs into one just embedding space. And then we can use this shared embedding space. We then input it into, we try to build some statistical model, and then we want to reconstruct the single-cell epigenomic reference part. And then meanwhile, when we just reconstruct this single-cell epigenomic part, and we can just generate the SE part for this given ST data. And so firstly, we apply some experiment firstly on the simulation data. So this simulation data is we just manually generate some spatial multimodal data. It means that for one spot, we both have the ATAC and the RNA. And so we just mask the ATAC modality of this simulation data. And then we just input this data into our model, and we get the inferred ATAC modality. And then we just compare this inferred one if it is just similar with the true one. So firstly, we just check the clustering result of this inferred spatial ATAC, and we find that the clustering pattern is quite similar with the original one with high NMI and ARI score. And then we also checked the region-specific Spearman correlation coefficient for some specific domains. In this case, it's the cluster 0 and the cluster 1. And then we find that the x-axis is the average accessibility counts of the true one, and the y-axis is the inferred one. And we can see that we can get a very high correlation, which means that the performance of our inferring model is very good. And then besides the simulation data, we also tested our model on the real world data, and which is the spatial RNA ATAC co-profiling data, and from the P22 mouse brain. And so, as we have mentioned about, we just we only use the ST part of this data, and then we just feed this reference and ST data in our model, and we can get the inferred ST data. And we do the validation between this inferred one and the true one. And we find that we check their region-specific correlation coefficient of the true and the inferred ST counts. And we can see that we slightly check that a different combination of our model architecture. And we can see even we apply different architecture of our model, and we can also get both the Spearman correlation coefficient and the Pearson correlation coefficient is just concentrate on a high level, which means that even in the real world data, our model can still, the inferred SC part of the SC data is still reasonable. And then we also checked that the shared embedding directly from our model, we can find that in this shared embedding, both the spot and the reference cells are just perfectly merged with each other, which means that some kind of the batch between the reference and the SD data is just removed. And that is very important to guarantee a good result. And then we also select some important transcriptomic factor and for some specific region. And then we just check if their transcriptomic factor binding site motif have some consistent enrichment in these regions. And we can see that the result is just very good. That we find that these two transcriptomic factor, their motif is just has a high enrichment in some correct specific region. And we also apply the clustering directly on the inferred spatial ATAC data for this P22 mouse brain case, and we find some new clusters that didn't appear in the original ST data, which means that our model, by using this kind of additional information, we can help to assist the clustering of the original ST data, and which also means that additional information is important to this ST data. And to make a summary is that we just design the deep model which inferred SE modality for a given ST data, and it shows high correlation coefficient in both simulation and real-world data. And it also shows the potential enrichment of spatial transcript binding site motifs. And it can also enhance the clustering result of the original ST data. And then for the future work, we want to try more complex ST data, for example, the tumor tissues, and we want to infer the other part, the SE part of this data, and check that if we can find some specific gene regulatory mechanisms for this kind of data. And we also want to, in this case, we just talk about the ATAC, and we also plan to infer the spatial histomodification, just using some different reference data. So because of time limitation, so if you want to know more details, please come to my poster. So that's all. Thank you.
I could spare you some more time if you want. But yeah, any questions? No, for Zhang, Weihang Zhang, for the audience. Yeah, please. If I have a suggestion for the organizers to have the mic maybe somewhere in the middle, it's easier for the
yes.
for the audience to come up to raise their questions. Yeah. I think a lot of courage to come to the front.
Thank you for insightful research. I'm not sure why the special cluster of using multi-omics is same that of using only attack sequence data.
You mean
you calculated the correlation between RNA and attack sequence data and only attack sequence data in first experiments.
You mean if I just do the correlation between the ATAC and the RNA-seq?
I think the correlation between RNA and attack sequence data and only using attack sequence data don't have to be same.
Oh, yes, it is actually because for our study, so you mean that the reference data and the given SD data, they should have from the same tissue. And that's why, and then for that single cell multi-omics data, the best choice is the paired data, which means that for one cell, we both know the RNA and for example, the attack or some other things. And in this, and then this SD data and this single cell multi-omics data, they are just sample from maybe different platforms, but they are still from the same tissue. And in this way, we can just find this two data, it has a high correlation, and that's why we can just use this kind of reference to fix that SD data.
Thank you.
Thank you for your question.
Yeah, I have some few questions, and the first that I really curious about if your model or in your future work is when we're using single cell ATAC sequencing or spatial ATAC sequencing, we want to discover some key components like these size regulatory modules, and can this model be performed to discover kind of things? Or
yes. So actually, the final goal is for the genomic research is to find some cis regulatory mechanisms.
Yes.
So our model is just we can provide how to say a computational way for you to generate another spatial epigenomic part for given SD data. And then if you get that as spatial epigenomic data, and then you can do anything you want, just using just maybe some TF binding site enrichment or you can get some peaks based on that spatial epigenomic, and then you can do anything you want just for some analysis of cis regulatory analysis. So we can just provide you the data to you, and then the downstream analysis is depending on you, and you can do anything you want.
Yeah, so I'm really curious about if we can.
Yes, here I show that it is an example. So here the right upper one figure is we just checked the TF binding site motif in our inferred data, and we find that it's really there are some important transcription factor, they are just motif enriched in some specific regions. So that's the maybe some important, can show some important things for some researchers.
Okay, thank you. Okay. Thanks for fantastic.
Thank you for your questions.
Any more?
So when we do data inferration, like using single-cell RNA-seq to infer single-cell attack, we see a lot of the predicted data sets always like different sparsity than the real one.
Yes.
I'm wondering, because you're predicting like spatial attack, are you having the same issue?
Yes, we have.
Okay.
So here you can see that in the red one, we use the statistical infer decoder. Here we select some, maybe the ZIP model. So this model just simulate the sparsity of the ATAC-seq. So in this way, we can generate some kind of sparse spatial epigenomic data. So I think this statistical model is very good